Imagine a world where your muscles simply give up during a workout, not from lack of will, but because of a fundamental cellular breakdown. This isn't just about fatigue. It's about a hidden molecular player that dictates your endurance. New research reveals that a protein called Ankyrin-B is a crucial and previously overlooked master regulator of muscle energy. But here's where it gets controversial. Could a deficiency in this single protein be undermining the benefits of exercise for some people? Skeletal muscle is far more than the engine of movement. It's a metabolic powerhouse, vital for regulating body temperature, maintaining the body's overall metabolic balance, and acting as the primary site for absorbing blood sugar after a meal or during exercise. When muscle function falters, it paves the way for devastating conditions like muscular dystrophies, age-related muscle loss, sarcopenia, and type 2 diabetes. This is why exercise, which enhances muscle function, is a frontline prescription for preventing and treating these diseases. However, the intricate molecular mechanisms that allow muscles to adapt to exercise and maintain metabolic health are still not fully mapped out. At the heart of this adaptability are mitochondria, the cellular power plants. Exercise triggers a remodeling of these mitochondria to meet energy demands. A key part of this process is mitochondrial dynamics, the continuous cycle of fusion, joining together, and fission, splitting apart. Fission is particularly important as it helps remove damaged mitochondria and allows the network to adapt. A protein called DARP1 is the primary molecular machine that pinches mitochondria apart to execute fission. This is where Ankyrin-B, ANK-B, enters the story. ANK-B is a ubiquitous scaffolding protein known for organizing structures within cells and acting as a transport adapter for organelles, including mitochondria in neurons. Previous studies linked deficiencies in ANK-B to metabolic syndrome, including obesity and insulin resistance, primarily due to its role in fat cells. Other research suggested ANK-B might be important for maintaining the structural integrity of muscle fibers. However, its specific dedicated role within mature skeletal muscle remained a mystery. To solve this, scientists created a unique mouse model where the ANK-B gene was specifically deleted only in skeletal muscle from embryonic development through adulthood. The results were startling. While these mice had normal body weight, body composition, and blood sugar control, even on a high-fat diet, they displayed a dramatic reduction in their capacity for endurance exercise. On a treadmill test, the ANK-B deficient mice reached exhaustion much faster and couldn't achieve the same top speeds as their normal counterparts. They also accumulated blood lactate, a marker of fatigue, more quickly. This pointed to a clear and specific problem. ANK-B is essential for optimal physical performance. Delving deeper, the research uncovered the root cause. The mitochondria in the muscle cells were broken. But not in the way you might think. They weren't fewer in number, they were the wrong shape. In the absence of ANK-B, the mitochondria became abnormally enlarged, hyperconnected, and failed to undergo proper fission. Think of it like a power grid where small, efficient local plants are replaced by a few massive, inefficient megastations. This dysfunctional network was under oxidative stress, even at rest, and critically had a severely reduced ability to burn fatty acids, the primary fuel for endurance exercise. And this is the part most people miss. ANK-B isn't just a passive observer. The study discovered that ANK-B physically interacts with the core fission machinery, directly binding to both DRP1 and its mitochondrial receptor, MFF. It acts as a master organizer, recruiting and clustering the fission proteins at the right place and time to efficiently execute mitochondrial division. Without ANK-B, DRP1 recruitment is impaired, leading to the hyperfused mitochondrial network. The plot thickens further. ANK-B deficiency also caused a disorganization of the sarcoplasmic reticulum, SR, the cell's calcium storage system, and reduced the critical contact points between the SR and mitochondria. This disrupted calcium signaling, a key regulator of both muscle contraction and mitochondrial function. This dual role of ANK-B in both organizing fission proteins and maintaining cellular structure suggests it synergistically coordinates the entire energy production process during metabolic stress. So what does this mean for us? This research fundamentally changes our understanding of muscle metabolism. It positions Ankyrin-B as a novel essential regulator of mitochondrial fission, specifically required for meeting the energetic demands of endurance exercise. While its loss didn't cause severe metabolic disease on its own in this model, the resulting exercise intolerance could significantly hinder the therapeutic benefits of physical activity. This raises a provocative question. Could variations in the Ankyrin-B gene explain why some individuals struggle with endurance training or experience excessive fatigue despite their best efforts? What's your experience? Do you think our genetic blueprint plays a bigger role in our fitness potential than we currently acknowledge? Share your thoughts in the comments.